BTEC Applied Human Biology, Unit 1, A1, Cells and Magnification Summary. So the equation to calculate magnification is magnification equals image size divided by actual size, or I am. And I is always the thing that you actually measure with a ruler. However, make sure you measure it in millimetres, because quite often you have to turn it into micrometres. So if it's 3 centimetres, for instance, that's 30 millimetres. You then have to times that by 1,000 to get 30,000 micrometres. What is the magnification of the diagram? If the actual thickness of the leaf below is 2,000 micrometres, but the image size of the leaf in the diagram is 50 millimetres. So 50 millimetres is 50,000 micrometres. So then you would just do, so that is the actual is 2,000 and the image is 50,000. So you do image over actual be 50,000 divided by 2,000, which is 25. In the magnification equation, image size is the part that you actually measure with a ruler. Microscopes, light microscopes and how they work. Light passes up through the specimen and is focused by the objective and eyepiece lens. Sometimes staining is required, and the advantages of light microscopes are they are cheap and portable and you can view living specimens. The disadvantages of light microscopes are they have a lower resolution than electron microscopes. The resolution is limited by the wavelength of visible light. The lower maximum magnification of times 1,500. So if you look at a microscope, an optical microscope, you have the eyepiece lens, you have the objective lens, which is usually times 4, times 10 and times 40. You've got the stage, which is where the slide is placed, the condenser to change the light intensity, the light source, the fine focusing wheel and the coarse focusing wheel. How transmission electron microscopes or TEMs work. Electromagnet focuses a beam of electrons through the specimen. The denser parts absorb more electrons, so appear darker. The tissue is fixed in a resin, then thinly sliced. How scanning electron microscopes or SEMs work. A beam of electrons scans over the surface of the specimen. Reflected electrons form an image. 3D images are produced on a computer screen. Advantages of electron microscopes. They have a higher maximum magnification of approximately times 500,000 and a higher maximum resolution of approximately 1 to 5 nanometers. Disadvantages of electron microscopes are the specimen material must be dead as it is placed in a vacuum. The image is produced in black and white, although it can be artificially coloured by a computer afterwards. Here is a transmission electron micrograph of an animal cell. So you can see that it is a cross section through the specimen and the darker parts are the denser parts. So you can see you've got the nuclear membrane, mitochondria, very, it's a bit blurred, this diagram. Mitochondria here, cytoplasmic membrane, Golgi apparatus, smooth endoplasmic reticulum, etc. Transmission electron micrograph of a mitochondria. So you can see the cristae, the infoldings of the inner mitochondrial membrane. Scanning electron micrograph of blood cells. So you can see that they are a scan of the surface and therefore they are in 3D. So you've got red blood cells here. Resolution definition. Resolution is the ability to see individual objects as separate entities. So it's the ability to distinguish between 2 points. Magnification definition is how much bigger the image is than the specimen. Why electron microscope resolution is better than a light microscope. Electron microscopy offers a better resolution than light microscopes and can reveal the structure of smaller objects not usually seen by the latter. This is because the wavelength of electrons is up to 100,000 times shorter than the wavelength of visible light of photons. Organelles. So, you can see here that we don't just call this the nucleus anymore. The dense part in the middle is called the nucleolus. And then the main part of the nucleus contains chromatin, which is DNA wrapped around proteins. You've got the nuclear envelope, a double envelope, and every so often you have little gaps which are called nuclear pores. Sometimes if it's like an epithelial cell, they might contain microvilli. They might show a centrosome with two centrioles close to the nucleus. A lysosome, are they just these circles? Mitochondrion with the inner foldings of the inner membrane. Rough endoplasmic reticulum has ribosomes dotted on it. Got the Golgi vesicle, which are stacks of cisternae on top of each other with pinches of Golgi body is the stacks of cisternae, and then every so often you'll have a pinch off, which is the Golgi vesicle. You've got the microtubules, ribosomes, the site of protein synthesis, cell surface membrane, cytoplasm, smooth endoplasmic reticulum. So eukaryotic cells contain a nucleus and other organelles that are bound by membranes. The plasma membrane is a selectively permeable phospholipid bilayer forming the boundary of the cells. The cytoplasm is a jelly-like fluid inside the cell in which the organelles are suspended. Chemical reactions occur there. The nucleus is surrounded by a nuclear envelope and has nuclear pores to allow movement of molecules through it. The nucleus contains chromatin. The function is to regulate cellular activity. Nucleolus is found inside the nucleus and produces ribosomes. Rough endoplasmic reticulum is the region of the endoplasmic reticulum that is studied with ribosome and engages in protein modification. Smooth endoplasmic reticulum, or SER, is an endomembrane system where lipids are synthesized, calcium levels are regulated, and toxic substances are broken down. Golgi apparatus, a system of membranes that modifies and packages proteins for export by the cell. Vesicles are small membrane sacs that specialise in moving products into and out of and within a cell. Lysosomes, an organelle containing digestive enzymes. ATS ribosomes are larger ribosomes found in eukaryotes, the site of protein synthesis. Mitochondria is the site of aerobic respiration, and they have two membranes. The inner membrane is highly folded to form cristae. The central part is called the matrix. Centrosomes and centrioles, organelles responsible for forming and anchoring the spindle fibres.